This video is part 2 in a 3-part series showing a day in the life of a field monitoring officer. This video will be focusing on ecology kick sampling. I'm now going to show you the equipment that we use for each sample. This includes a set of shears to cut through any overgrown areas. Then our PPE, which includes a life jacket and waders. We use a clean set of waders for each site, so we carry multiple pairs. I have actually just used these to collect the water sample, but we have to ensure that they're clean before getting into any site. A stopwatch so that we can accurately record the 3 minutes that we require a kick sample done for. Sampling pole, again we have multiple of these in the van to ensure that we're using a clean one for each new site of the day. Sampling tray to decant the sample into and have a look through, including some water in a squirty bottle to help clean through the net. Sample pot for putting the actual ecology sample into, and then a diatom tube and toothbrush which has to be clean, and I'll run through the use of that later on. And then a YSI meter to record the conductivity of the water, and then most sites require a water sample as well, which I've already collected for this site. And I'll now show you an example of how we take a kick sample. The process for most sites for ecology kick sampling is very similar, but there are slight changes that we have to make depending on the specific site. So as this is quite a small stream with a silty bed and not much flow, I'm holding my net quite close to my foot and I'm stamping up and down to agitate the invertebrates into the net. On a bigger stream or something with a gravel bed and higher flow, I'd have the net further back and I'd be kicking more vigorously to disturb the invertebrates and allow the flow to push them into my net. Regardless of the specific site, the general process for ecology kick sampling is very similar. Here I'm just looking through my net to ensure that I haven't missed any invertebrates that need to be added to the sample. So earlier on, when running through the equipment list, I mentioned this diatom tube, and essentially this is a tube that's filled with a preservative. And what we'll be doing is we'll be collecting 3 cobbles from the stream, which will be covered in diatoms, and we'll then be using a clean toothbrush to scrape these diatoms off, and we'll be putting that into the preservative, which will go to the lab to be analysed. Whilst doing this, we'll also be doing a manual search on the stones, looking for any invertebrates, and if there are any, we'll be adding that into our ecology pot. So I now have my water sample, my ecology kick sample and my diatom sample. I'm going to pack up my kit, head back to the van and move on to my next site for today. So I'm just now walking across to the next sample site. I've got a fresh net and a fresh set of waders, all of the same kit as before. This one is a river rather than a stream, but the principle is the same. So I won't go into too much detail with what I'm doing because it's very similar. However, I will have a bit of a chat just about some slight differences that you need to consider at different sites. So as I mentioned just now, whilst the principle for a river is the same, there are different considerations that we need to make. On each site, we're required to fill out a list of questions regarding the different habitats and features within the watercourse. With the last one, there wasn't too much to it. It was a very straight, small stream, whereas this one, we do have a little bit of feature to it. So we have some reeds further up stream. We have a riffle, and whilst it's generally quite an even depth, just after this meander here, we do have quite a deep section. So what we'll be doing is we'll be having a look at the site before we start, and we'll be looking at each different habitat type. And what we'll be doing is we'll be breaking down the 3 minutes proportionately to the different habitats that we have. Every site requires a list of questions to be answered regarding the site itself. So the sorts of questions that we need to answer are whether we've seen any protected species or any non-native species, such as an American signal crayfish. And we'll also be having a look at the different habitats that I discussed just a moment ago, and we'll be recording what sort of percentage of the substrate is gravel, sand, silt, that sort of thing, just to give us a good idea of the sort of site that we're looking at, and any effects that those habitats or substrate types may have on the invertebrates that we find here. So I'm just going to do that now, pack up my kit and head back to the van, and then tomorrow what I'll be doing is going to a different site with some new kit, and I'll be doing a sweep sample, which is a type of sample that we do when the site is too deep or inaccessible for us to actually get into the channel. So it's a new day and I'm on a new site, and as I explained yesterday, there's a different type of ecology sampling that we do, which is called sweep sampling. And this is what we do for any sites that are inaccessible or perhaps dangerous to get in the channel. So whilst this site doesn't look too bad to get into, it's actually full of silt and therefore wouldn't be safe for us to get in. So what we'll do is we'll be sampling this with an extension on the end of our net, so that we can sample this from the bank. And what we'll be doing is we'll just be working our way upstream, prodding the bottom with the net and trying to agitate the substrate and get the invertebrates in that way. So I'll just give a quick example of how that looks. Hopefully that gives you a good idea of the difference between a kick sample and a sweep sample. I'm actually going to do it properly now, and it's the same as with the kick sample. It's a 3-minute sample whereby I'll be assessing the whole length of the site, and I'll be making sure that I split up the time that I'm sampling proportionately to the different types of habitats. To be quite honest, there's not a lot to this site, so it's going to be a fairly even run up. But once I finish that, as with the kick sample, I have to fill out the same sorts of questions as before. It is obviously quite a bit harder because I'm not in the channel and I can't feel the substrate. So what I need to do is just have a good look up and down the site, and make sure that I'm having a look for any different habitat types and substrate types and any different effects that it may have on the invertebrates that live here. At the end of each ecology sampling day, when we get back to the depot, we need to use industrial methylated spirit to preserve the sample. This ensures that when the analysis and reporting team receive the sample, all of the invertebrates that we've gathered remain intact and they're able to identify and quantify everything that's been found. I briefly mentioned the biosecurity procedures earlier on, whereby we always use a new set of waders and a new set of nets at each ecology site. The final thing that we need to do at the end of each ecology day is ensure that we leave our equipment somewhere to dry and ensure that it is left for at least 48 hours before it is used on a different site. The other 2 points in the check, clean, dry methodology, i.e. check and clean, must be carried out on site before leaving the site. This is to ensure that any organisms found on our equipment before we leave remain on the specific site that we were at and aren't carried and spread to a new location. As a field monitoring officer, we visit multiple different watercourses across the UK. It is therefore of utmost importance that we strictly adhere to our biosecurity procedures. This pretty much sums up the average ecology sampling day, but if you wish to find out more, please visit the About Us section on the National Monitoring SharePoint page. The next video in the Day in the Life of video series will be showing the other main type of work that we do in the field monitoring team, and that is electrofishing surveys. Thank you very much for watching this video.